Welcome to the Longevity Papers podcast.
Hi, everyone.
This week, we're plunging into a collection of fresh research, some just published, others hot off the preprint servers.
Yep, and it's packed with insights specifically relevant to you, the biotech researcher, working on extending human health span and lifespan.
Our aim today is to quickly dissect the core findings from these sources, pinpoint what truly moves the needle for longevity science, and, apply a critical lens to the data.
Absolutely. Staying curious, staying honest, and really focusing on the practical implications.
Exactly. We've got a mix covering systemic interventions, cellular therapies, some novel diagnostics, and, deep dives into aging's fundamental mechanisms.
Sounds good.
So let's jump right in and unpack a study exploring a combination therapy first.
Okay, yeah. This first paper comes from Gicchioni and colleagues. We actually picked it up via the longevitypapers.com aggregator.
Ah, okay.
And while this specific journal wasn't right there in the snippet, the central finding is really compelling. Combining trametinib, which is a MEK inhibitor, with rapamycin, the mTORC1 inhibitor, it shows additive benefits in mice for both lifespan and health span.
Okay, so targeting two different critical pathways, MEK and mTORC1. That's a strategy we're seeing more interest in for complex conditions like aging.
Definitely.
What kind of lifespan extension did they actually observe?
So the lifespan results were, highly statistically significant in their mouse model. In the female mice, the combination significantly extended median lifespan by 34.9%.
Wow, nearly 35%.
And maximum lifespan by 32.4% compared to controls.
Okay.
For males, it was still really impressive. 27.4% increase in median and 26.1% in maximum lifespan. P-values less than 0.0001.
Those are very substantial increases. How did the individual drugs stack up on their own?
Yeah, good question. Rapamycin alone was quite effective, too. It increased median lifespan by around 17% in both sexes.
Okay, still significant.
Right. Trametinib alone had more modest effects, maybe 7% in females, 10% in males. So the combination clearly outperformed the individual drugs.
Right.
And the analysis indicated this was an additive effect, not necessarily synergy, but, still very valuable.
Yeah, additive effects from distinct pathways are still powerful.
Yeah.
It suggests you don't necessarily need perfect synergy to see enhanced results if you hit multiple nodes, right?
Exactly.
So beyond just living longer, what did they find regarding health span? You mentioned that too.
They looked at several key markers. Tumor incidence, for instance, was notably reduced. Now, that's maybe not entirely surprising given rapamycin's known effects.
True.
But still important. Liver tumors, for example, dropped from a high of 50-70% in the control groups down to maybe 10-20% with the combo therapy in both sexes.
Okay, that's a big drop.
Yeah, and spleen tumors were also significantly reduced, especially in the male mice.
Reducing major age-related pathologies like tumors would certainly contribute to extended health and lifespan, at least in mice. What else did they examine?
Brain health was another focus. They used PET imaging and found the combination treatment actually prevented the age-related increase in brain glucose uptake in female mice.
Oh, interesting. In which areas?
Across multiple regions, including the striatum, cortex, and cerebellum. And, changes in brain glucose metabolism are often seen as part of the aging process.
Right. And what about inflammation? That's another big hallmark of aging.
Yep. They looked at that too. They saw reductions in brain inflammation markers, things like activated microglia and astrocytes, particularly in the striatum.
Okay.
And peripherally, outside the brain, there were decreased inflammation scores in kidney tissue and lower levels of various pro-inflammatory cytokines in the blood plasma. Things like TNF, IL-17A, IL-23R.
So a pretty broad anti-inflammatory effect then.
It seems like it, yeah. And that likely contributes significantly to the overall health span benefits they observed.
So impacts on cancer, brain metabolism, inflammation.
Mm-hmm.
Did they manage to explore the molecular mechanisms behind this combined effect? How was it working?
They did. They used RNA sequencing, and it revealed that the combination treatment induced some unique transcriptional changes patterns that weren't simply the sum of the changes from the individual drugs.
So maybe hinting at something more than just simple addition at the molecular level.
Potentially. A really prominent feature was the downregulation of inflammation-associated genes, which, lines up nicely with the observed reduction in those inflammatory markers we just talked about.
Understanding how these different targeted pathways interact molecularly is so key for designing rational combinations down the line. Okay, now let's put on our skeptical hats. What are the caveats or potential downsides here?
Well, a really significant point is that the side effects commonly associated with rapamycin.
Right, the metabolic ones.
Exactly. Things like decreased body weight, hyperglycemia, some liver lipidosis, and even testicular degeneration in older mice. Those were still present with the combination therapy.
Okay, so the combo didn't mitigate those.
No, it didn't seem to. Trametinib alone didn't cause these issues. So for any translation to humans managing these metabolic side effects, that remains a major challenge.
That makes sense, since rapamycin is still in the mix. What about the trametinib itself and those brain findings you mentioned? Any nuances there?
Yes, actually. They noted that trametinib barely crossed the blood-brain barrier at the dose they used in the study.
Oh, really?
Yeah, which implies that the effects they saw on brain glucose uptake and brain inflammation might actually be indirect.
How so?
Well, possibly mediated through the reduction of peripheral inflammation, which then influences central nervous system processes. So it's an important detail when interpreting that brain data.
Got it. So a powerful demonstration of potential additive effects, but the known trade-offs, especially from rapamycin, persist. For you, the biotech researcher listening, what's the key takeaway from this paper?
I think it really reinforces the value of combination therapies targeting distinct fundamental aging pathways like MEK and mTORC1. Showing additive effects are achievable by hitting different nodes, while that provides solid support for multimodal strategies.
Right.
It also offers some molecular data on how these specific pathways interact, which could inform future designs. But again, that side effect profile highlights the ongoing translational challenges we need to overcome.
Okay, clear. Moving on from systemic drug combinations, let's switch gears a bit to cellular therapies. We have an interesting paper here on hematopoietic stem cell transplantation, right?
Yes, this is a study from Kontarek-Sisla, Zhang, Karazi, and Breiter. It was just published in Nature Communications on June 2nd, 2025.
So very recent. What's the core claim?
Their main point is that a non-genotoxic approach to hematopoietic stem cell, or HSC, transplantation can rejuvenate the blood and immune system. And importantly, it can mitigate age-related blood disorders in mice using young donor HSCs.
Non-genotoxic, that's the really critical part there, isn't it? Traditional HSC transplants often need pretty harsh chemotherapy or radiation for conditioning.
Exactly. Which is toxic, especially for older or more frail individuals, the very people who might benefit most from rejuvenating their blood system.
So what's their gentler approach?
The novelty here is their conditioning regimen. They used antibody-mediated depletion of the host HSCs with something called CD45 SAP.
Okay, what's that?
It's basically an antibody that targets CD45, which is on blood cells and their precursors linked to a toxin, saporin. So it selectively kills off those cells.
Gotcha.
And they combine that with a mobilization strategy using GCSF and AMD 3100 drugs, often used to push stem cells from the bone marrow into the blood. The whole idea was to clear space for the young cells without that harsh toxicity.
And did it work? Did this gentler method actually allow the young cells to engraft in aged mice?
It did, quite successfully.
Yeah.
They achieved significant long-term engraftment of young HSCs in mice that were 16 months old, so already getting on in age.
What kind of engraftment levels?
They saw substantial contributions from the donor cells in the peripheral blood, something like 60% of myeloid cells, 40% of B cells, and around 30% of T cells were donor-derived long-term.
That's pretty significant chimerism.
Yeah, and in the bone marrow itself, they recovered about 4 times more donor HSCs compared to just using the antibody depletion method alone.
Okay, so the young cells took hold and started producing new blood cells. What about the impact on rejuvenation markers? Did the blood system look younger?
Yes, it seems so. The young HSCs effectively reconstituted the hematopoietic system and improved the T cell profile towards a more youthful state. For example, about 40% of naive CD4 plus and CD8 plus T cells were donor-derived,
Compared to what in the host cells?
compared to a pretty low percentage, maybe 5–15% from the aged host cells still remaining. So that directly addresses a key aspect of age-related immunosenescence, that decline in naive T cells.
Makes sense.
They also saw elevated hemoglobin levels, suggesting better red blood cell production.
Restoring youthful immune function and blood production. That sounds like a direct way to tackle some age-related blood diseases.
Right.
Did they test this therapeutically?
They did. They used a mouse model for myelodysplastic syndrome, MDS, which is a kind of age-related blood cancer.
Right.
And in this model, their transplant strategy significantly reduced the incidence of these hematological malignancies. It dropped from 75% in the untreated mice down to 33% in the transplanted group.
Wow, less than half.
And maybe even more crucially, it completely prevented the progression to acute leukemia. That happened in 25% of the untreated mice, but 0% of the transplanted ones.
Preventing fatal blood cancers, that's clearly a major health span benefit. Okay, critical lens time again. What are the limitations or challenges here?
Well, the primary impact shown is really focused on rejuvenating the hematopoietic and immune systems and preventing those specific blood disorders.
Right.
While that definitely improves health span and likely median lifespan in those mice, the direct impact on maximum lifespan across all causes of aging, well, that's less clear from this particular study.
Fair point.
The authors themselves acknowledged some limitations too, like they didn't formally test the long-term self-renewal capacity through serial transplantation, which is kind of a gold standard for stem cells.
Right.
And they didn't do direct functional immune assays, like testing how the rejuvenated immune system responds to an infection or a vaccine challenge. They also noted some potentially small sample sizes in certain experiments.
Okay, important considerations. And what about the big picture? The translational hurdles for getting this kind of therapy into humans?
Oh, they're significant for sure. First, you need a source of young, healthy human HSCs. For elderly recipients, that's a challenge. Might require allogeneic donors.
Which comes with risks like graft versus host disease?
Exactly, although maybe the gentler conditioning helps mitigate that somewhat. Or, looking further ahead, maybe iPSC-derived HSCs could be an option one day.
Right, but that's still developmental.
Very much so. Then you need to develop a safe and effective antibody-based conditioning regimen that works reliably in humans. That's a major R&D effort.
Yeah.
And finally, just defining the clinical endpoints for using this as a preventative therapy in otherwise asymptomatic elderly people. That's complex from both a regulatory and a clinical trial design perspective.
So it sounds like a really important proof of concept for a safer approach. And it shows clear potential against age-related blood decline and disease, but still a long road ahead for actual clinical implementation.
That sums it up well. But still, for you researchers out there targeting age-related hematopoietic and immune decline, this offers a tangible path towards potentially safer cellular therapies. It could significantly improve health span and quality of life for the elderly down the line.
Absolutely. It's a vital piece of the aging puzzle. Okay, so we've done a deeper dive into those two key papers, but you mentioned this week brought a whole flurry of other interesting research across various fronts in longevity science. Let's maybe briefly highlight some of those other notable ones.
Yeah, absolutely. There was quite a bit. For example, over on bioRxiv, remember that's pronounced bioRxiv.
Got it, bioRxiv.
On June 4th, Salignon and colleagues released details of a new transcriptomic aging clock they're calling PASTA.
PASTA. Okay, another aging clock. We see a lot of these. What makes this one potentially notable?
Well, they claim it's platform and tissue agnostic. If true, that would be a major advantage for wider use and comparing results across different studies and systems.
Yeah, that would be useful.
And their analysis using this clock pointed towards mitochondrial translation and mRNA splicing as really key processes linked to aging and rejuvenation.
Interesting targets.
Yeah, so it suggests potential new therapeutic avenues and also provides, another order tool to evaluate interventions. But again, it's a preprint, so it needs validation. Still, the potential's intriguing.
Definitely. And sticking with new tools and fundamental mechanisms, there was another bioRxiv preprint, this one from Kriz et al. On June 1st, about somatic mutations in the aging brain.
Right. This study introduced a really powerful new method they call duplex multiome.
Duplex multiome, what does it do?
It allows for highly accurate detection of somatic mutations, those genetic changes that accumulate in our body cells over time.
Okay.
And crucially, it links those mutations to gene expression changes within the same single cell.
Oh, wow. That's powerful.
It really is. Using this, they mapped out distinct patterns of somatic mutations accumulating in different brain cell types with age. It's a significant technical step forward for understanding how these spontaneous mutations might contribute to brain aging.
Very cool. And shifting topics slightly, what about fibrosis? That's such a common age-related problem.
Yes, Grow et al. Published a paper in Advanced Science on May 30th about a molecule called nephronectin, or NPNT.
NTNP?
They identified NPNT as potentially having anti-aging effects. They noted it's reduced in age-related idiopathic pulmonary fibrosis, or IPF,
Okay.
and it appears to influence cellular senescence through a specific pathway involving ITGA3 and YAP1. What's really interesting is they showed a small molecule called essin.
As in, like from
What?
horse chestnut.
That's the one, yeah.
Uh,
They showed essin could elevate NPNT levels and actually reduce fibrosis in mouse models. So this suggests NPNT and its pathway could be new targets for tackling age-related fibrotic diseases.
Fascinating. And was there another paper focusing specifically on cellular senescence itself?
Yes, Wang et al. Were in Nature Aging on May 31st. They developed something called the Human Universal Senescence Index, or HUSI.
AGUSI, what's the goal there?
They developed a robust transcriptome-based index, so based on gene expression, designed to identify senescent cells reliably across various different tissues and conditions.
Why is that important?
Well, because senescent cells aren't all the same, there's a lot of heterogeneity, so having a better tool to characterize that heterogeneity and track their accumulation is really crucial for research and also for evaluating senolytic therapies as they hopefully move towards the clinic.
Makes sense. Better measurement tools are always needed. Any new genetic links to human longevity reported this week?
Yes. Baghdadi et al., published in Geroscience, also on May 31st. Looking at human genetics, they found some rare genetic variants within the MAPKIRK signaling pathway that were associated with human longevity.
The Jap-Kirk pathway. Okay.
And they did functional follow-up work in mouse cells, showing these specific variants actually altered the signaling cascade. So it directly links specific genetic differences in this pathway to lifespan regulation.
Interesting. And mitochondrial health, that's always a big theme in aging research.
Absolutely. Nuzhu et al. In the journal Aging, on May 30th, looked at a small mitochondrial protein called ROMO1.
ROMO1.
They showed that overexpressing ROMO1 actually protected sensitive cysteine residues within mitochondrial proteins from damaging oxidation.
Protecting against oxidative stress specifically in the mitochondria.
Exactly. And this improved mitochondrial function and, importantly, extended lifespan in C. elegans. So it reinforces that concept that maintaining redox balance specifically within mitochondria is a viable longevity strategy.
Okay. And maybe one more. Was there a review highlighting a potentially broad therapeutic target?
Yes, there was a review referenced as Source 85 in our notes that made a strong case for looking at necrosis differently.
Necrosis, like uncontrolled cell death.
Right. Often seen as just messy, accidental cell death. But this review argued it's not merely a consequence of damage, but potentially a fundamental driver of biological decline and aging.
A driver, not just a result.
Exactly. And the review proposed that specifically inhibiting necrosis pathways could be a significant, perhaps even game-changing strategy for treating a wide range of age-related conditions and maybe even slowing aging itself.
As a provocative idea.
It is. We also saw that preprint from Shen et al. Looking at tirzepatide.
The diabetes and weight loss drug.
Right, showing it reduced diet-induced senescence and liver fibrosis in mice, which again underscores how these metabolic interventions can impact specific cellular aging processes in different organs.
Okay, wow. That's quite a range of research just from 1 week. Let's try to connect the dots across these findings. What does this week's collection of papers really signal for you, the biotech researcher listening in?
Well, what really stands out to me is the multifaceted nature of the progress. We're seeing movement on systemic interventions like those drug combinations, advancements in refining cellular therapies like the non-genotoxic HSC transplant,
Right.
and these crucial steps forward in developing better tools to measure aging and understand the underlying mechanisms like the new clocks, the senescence index, that single-cell mutation analysis.
It really feels like it's not just incremental progress in isolated silos anymore, doesn't it? The breadth of these approaches suggests that aging is being tackled simultaneously from genetic, cellular, tissue, and systemic levels.
I agree. And the development of more precise tools to assess biological age and cellular states, like these transcriptomic clocks or senescence indices, that's becoming increasingly vital for rigorously evaluating the efficacy of potential interventions.
Yeah.
You need good measurement.
Absolutely.
This collection also, I think, underscores the potential power, but also the complexity of combination approaches, whether that's combining drugs targeting different pathways like the first paper, or maybe even potentially combining different types of interventions down the road, like a cellular therapy alongside a metabolic intervention or a senolytic.
Yeah, the possibilities start to multiply. But as always, we need that healthy dose of skepticism, right?
Always. Mouse models, they provide essential insights, but translating those findings to complex human physiology, That's a significant challenge, particularly regarding efficacy and managing side effects, as the combination therapy study really highlighted.
And we need to remember, those preprints on bioRxiv offer exciting early access, but they haven't gone through the full scrutiny of peer review yet. Validation is absolutely key.
Couldn't agree more.
And the translational hurdles we discussed with the HSC paper, things like sourcing materials, developing human-compatible protocols, defining clinical endpoints, those are challenges that apply pretty broadly across the field, wouldn't you say?
Oh, absolutely.
How do we take these promising lab results and actually develop them into safe, effective, and importantly, scalable therapies for aging humans, especially for the elderly population who could see the most profound benefits?
Those really are the central questions shaping the field's trajectory right now. It's where the rubber meets the road.
So, thinking about that path forward, what does this all mean? You mentioned the potential for additive effects with combinations, and we know aging is incredibly complex, involving multiple interacting processes.
Right.
Really does seem like designing and testing complex, multimodal longevity interventions is the next frontier. What do you see as the biggest opportunities and maybe the biggest challenges in actually doing that? Testing integrated strategies, maybe hitting cellular senescence while boosting stem cell function and optimizing mitochondrial health all at once.
Well, opportunity is huge, right? Targeting aging from multiple angles could yield potentially synergistic or at least significantly additive benefits that single interventions just can't achieve.
Right, a bigger impact.
But the challenge lies in the sheer complexity. How do you even design the preclinical studies for multi-component therapies effectively?
Yeah.
How do you navigate potentially incredibly intricate clinical trials? What are the regulatory pathways for interventions targeting aging itself rather than single diseases? Not to mention the manufacturing and delivery logistics for potentially complex therapies. It really demands a new level of integrated thinking, from basic science all the way through to clinical application and delivery.
Definitely food for thought there.
Oh.
Okay, that wraps up this week's deep dive into the latest longevity papers. We hope this has provided you with some valuable insights and maybe sparked some new ideas for your own research.
Remember, the journey to understand and intervene in aging is, well, it's a really dynamic field. There's exciting progress happening constantly, every week it seems.
Thanks for joining us. Until next time.